Yes. Okay.
Oh, happy birthday.
Hi everyone. Hi everyone. Welcome to Pint of Science. Welcome to this last waiting for Pint 21 event because no, waiting for sorry, waiting for Pint 22 because our next event will actually be the festival that will take place in May from the 9th to the 11th. Just a quick reminder. So tonight we have a change of plans. So we will start with the presentations and we will hand the quiz at the end of the night. Before starting, I would just say a big thanks to all the guys, but I will present them later. All the guys of the team that are working and make everything possible here, and of course all the speakers that will that join us tonight. And I want to thank our co-host for tonight, that is Simula. We will have time to present Simula better later. But now I would, before introducing the first speaker, I would just remind to everyone that we are also streaming on Facebook, on YouTube, and LinkedIn. Yeah. So everyone who is following the streaming can also send questions, and we can just write them down in the comments, and we can read them to the speakers. Our first speaker tonight is Nessie Hancock, right? Naji Hancock.
Thank you.
He has a cross-disciplinary educational background across electronics and cybernetics, molecular biology, applied statistics, and computer sciences. He has more than 40 years of experience in the computing industry and as an academician and researcher, almost always in parallel. He is the co-founder and CEO of InVirtualis, a company working in computer-aided molecular design, which results heavily on modeling and simulation. He's a nerd who can focus terribly on something and forget other things. He was not able to stay long with us today. I'm sorry for that, because he forgot that actually today is the birthday of one of his best friends, so he will leave earlier. But please welcome Naji, and please, the stage is yours.
Thank you. There's my presentation. My button. That wasn't meant like that. Sorry. All right. As you heard, my name is Naji, and there is in one of the slides the correct pronunciation. I will try to talk about mathematical, statistical, and computational modeling. Of course, bringing the whole thing to AI, because that's what everybody else is talking about. I just want to take you through the journey that I have experienced. These ladies, very kind ladies, didn't believe how old I am. I am 70 years old. And I'm sort of the history of computing science. I started very, very early, and I've been in both the academia and in the industry for more than practically about 50 years now. So I'm going to take you through the story. Let me see this one. It's first a bit, the agenda is a bit about me, a bit about InVirtualis, our startup, a little company that we're very proud of. And I'll try to say a couple of things about how we learn mathematics and how we should learn mathematics. Remember, I've been not only in the industry, but lecturer still, basically, at the university, both at the University of Oslo and OsloMet for quite some time. So I've seen how we're not teaching them, basically. I'd like to just mention that because that's one of my hobbies. And then I'll talk about modeling. Of course, a very, very simple version. I'm not going to drown you in formulae, but I will try to give you an understanding of how we use them, amongst other things, and mathematics in modeling. So we'll be talking about the deterministic paths, which is the analytical paths, which is like polynomials and God knows what, kind of algebraic equations, basically. And then I'm going to try to jump over. I have this little bridge in between them. One little example. We'll jump over to stochastic modeling, random modeling, statistical modeling, and then we'll look at computational modeling after that, which will bring us to artificial intelligence that builds upon these, basically. Me, that's me. I have, well, you've just heard. I'm relatively well educated, you can say. I have a little bit of computer science and cybernetics and molecular biology at the bottom, and then applied statistics and computer sciences on top of all of that. So if you're interested to talk about me, I am usually very interested about to talk about myself, so you can talk with me afterwards. I'd like to say a couple of things about the company as well, so that you understand a little bit where we're coming from, what interests me today. The company is called In Virtualis. It stands for in the virtual. The Latin speaking can confirm, I hope. It is in many ways a complement of what is called in vitro, in the class, basically, lab experiments, and in vivo, in living things. This is in the virtual. So what we're trying to do is create virtual laboratories that just move the laboratory into the computers, if you want, let's be roughly speaking. We're trying to create an intelligent digital twin of the molecular world, which means that we are using these kind of techniques ourselves. We use a little bit of modeling, a lot of modeling, and a good deal of artificial intelligence. We're doing that kind of stuff, plus other things. Okay, so what are we going to talk about? A model. The headings are gone somehow. It doesn't really matter. It's the title was model or modeling. What modeling is all about. This is something I like to repeat when I'm talking about modeling, because this is one of the things that I've learned around, when was it? Around 76, I think, when I met a guy called Kristen Nygaard, if you ever heard of him. When I came here, he was working, he was one of the gurus of computer science, basically. One of two who founded object-oriented paradigm. And he used to keep on saying, well, what we do when we program is abstract, create an abstraction of the world, and then build instructions around that. That's a model, basically. So a computer program is really a model of reality. And that's Professor Kristen Nygaard. He passed away. And he is one of the fathers of the object-oriented programming paradigm. Maybe you know or not. This is the point where I usually tell a tiny little, slightly dramatized version of history. It's not, I mean, it's not so dramatized. 10% is my imagination, if you want, just to make life a bit interesting, but the story is actually true. After the Second World War, when the war ended and people who shouldn't be here left, they were here also, not only for, but also for heavy water, if you maybe remember, because heavy water was essential for creating this nuclear experiments, basically, and the bomb, eventually. So they left, but the heavy water remained. because it was actually a byproduct of some other chemical process at Hydro Rjukan facility. Very good. So the Defense Department or the Defense Research Institute, FFI, Försvarets forskningsinstitut, started wondering what they were going to do with this. They had this, okay, well, it's obviously useful for something, since the Germans were so interested. So what can we do? Can we try to create a nuclear power plant, for example, and do experiments around it, or maybe even generate energy? So, yes, of course, good idea. So what do? We'll go ask our friends how we do this, how we calculate the rod dimensions, how much they should be in heavy water. This thing needs some amount of calculation, obviously. So they went and asked the United States friends, said, friends, can you please give us the formulae for the a heavy water plant? And they said, France, you're so good, France. Not that good. So they didn't get it, basically. That's the drama part. Of course, they didn't. They probably taught some sort of a diplomatic language at that point. And so what happened was that FFI, the Research Institute for Defense Research Institute, employed this guy, Christian Nygård, who called in his friend, Julian Dahl, and they started looking at how we could first analytically formulate this. It didn't work out very well. It was difficult. So they said, let's simulate instead. Okay, we simulate. They sat down and started simulating. You can imagine how hard it is to simulate one particle going and hitting another particle, creating many other particles, which then you have to, on paper, if you're doing it manually, follow and sort of calculate the probability of that hitting another particle and creating other particles and all. That wasn't very doable manually. So they started programming it. And what they had was not much, Fortran 4. That wasn't a very efficient language. It was a formula translator is what it stands for. It was actually for scientific computing, made at that time, but still it was not sufficient. So they started to use a block structured language, if you know what that is, Algo, and that wasn't enough either. So what they did was they created a language in the end just for being able to simulate this nuclear crash into each other kind of events. That's what they did. And guess what that language was called? Quiz? Simulation language, abbreviated Simula. Simula Research Lab is that's where that comes from. So now you know where Simula Research Lab builds what it builds upon. It builds upon the findings of these guys. And they managed to, of course, both simulate the whole situation, create the first nuclear power plant in Halden, and in addition, they create the object-oriented paradigm with Java and C++, many other languages, Python, etc., etc. Is that me? Yes. It's usually me. Okay, we'll keep it outside. Yeah. Now that's the story. That's the history. Okay. And then, of course, a computer program is a model of reality. So, sort of backtracking a little bit from there, you can say that any mathematical, statistical, or computational formulation is also a model of something, some physical or abstract reality. Well, it's usually an abstract reality because you take the physical reality, abstract it, and then model it, typically. And who said that? That is one of the students of that guy. Me. Basically, I've been talking about it. And that led to me doing my PhD in modeling, amongst other things, and my master in modeling, etc. All right. Now, the title, which isn't there, sorry for that, says, I believe, modeling, analytical modeling or deterministic kind of stuff, or something like that. The first of the three kinds of modeling. Let's just stop and talk a little bit about how we teach mathematics. You show the kids something like this, and then you say, okay, if you do this and that mechanically, you sort of tell them how to solve this equation, if there is any solution to it, basically, right? You basically make them, we today, unfortunately, I'm one of the guilty ones. I didn't realize it until it was pretty late. We make it really difficult for the kids to learn because we're forcing them to memorize without understanding the basics of mathematics. And there is, you probably heard about that, it's many people start hating mathematics pretty early in life. And that's one of the reasons. But we could have tried to explain, that's how I believe we should teach mathematics. We could have tried to explain to these kids that there was this guy, like Euler, for example, and others. If you looked into the quiz, there are other names that are related as well. This guy tried to do something. He hit his head on the wall several times because the tools he had wasn't sufficient for doing that. It's a bit like the story of Olympiad and Kristen Nygaard discovering Simula or creating Simula, if that's what it is. You need to develop the tool for the job, often. And most often, when you're doing, trying to take some sort of reality and model it from nature, nature is in constant change. So one of the things that you will need is a differential equation because it shows you how today's value is dependent on yesterday's value and tomorrow's value is dependent on today's value. The changes, your modeling change. For God's sake, nature is change to a large degree. So when I start talking about modeling, I usually start talking about this part of modeling. There are many other venues of modeling, of course, but change is one of the things that we absolutely need to model. And it's one of the things that they hate, the kids, integrals and differential equations. No, thank you, basically. I don't know. This guy used to be one of my, it's on my website at the very top right. This, I claim that this is Pantere. Heraclitus is an old Greek philosopher. He said Pantere, that means everything flows, everything changes, basically. That, I believe, is the only truth that we can really keep to in the world. So we start from that. This is the model of from fluid dynamics. It says the flow in time changes with respect to some gradient and some attraction factor. It could be the gravity, if that is what it is, if there's nothing else that stops or alleviates the gravity, basically. That's the only thing it says. If you explain to the guys that something is flowing, and that is going to be changing with respect to what's pulling and pushing. This is one way of seeing a formula instead of just pushing it to people. And this is one of the examples of how we do analytical or discrete, deterministic, modeling, not stochastic. There are many other examples. We are still in the modeling deterministic part. This is just an example. This is the bridge I talked about that we're going to step across to stochastic modeling. And I have to be very fast in presenting because people are waiting for me. So I'm running through this. This is just another example. Imagine you have a time series, data in time. Sort of think of an x-axis and a y-axis, and then these spikes all over the place, giving some value at different times, right? And what you want to do somehow, maybe you're a musician because of that, or maybe you're about to create a new science, maybe something like probability theory, for example. Okay, so what you really want to do is take these values and sort of cluster them, group them, so that you can count how many times each value occurs. It's a bit back to Christian doing it on paper by hand. If your data set is large, and it does get larger and larger, is not a very good way of doing it. It doesn't help much. So what you do is you formulate something. You find a way of doing it through using algebra or something. One of the guys called Fourier, I believe he was French. I don't know if there are any other French people here. He decided that he could decompose a function that was in space or time into spatial or temporal frequency components, basically. And he formulated it. I'm not going to go through it today, but that is the kind of formulation that he was looking for. That's what we call the Fourier transform today. It's a transform. One of the nicest things about the Fourier transform, the way he thought about it, it's not only him. There were many others who started thinking in that manner earlier before him as well. One of the nicest things that you never hear about, maybe except from me, because I was very occupied with that kind of stuff. You have a problem in an algebraic domain, in an algebra, with certain operations and this and that. You can't solve it in that algebra. What do? You change the algebra. Basically, that's what he did. It's the same later on, things like you may have heard about the Laplace transform that builds upon this. It's the same kind of thinking, basically. You take one algebra, think of it as an algebra with its operations and operands and all that stuff, then change that. That's what they basically did. They moved the domain of the problem fully. And they created the Fourier transform. Where can it be used? Well, maybe if you like music, for example, if you like shredding, heavy guitar and all that stuff, you know this overdrive that you hear, the sound, very harsh sound. That is actually one way you can visualize what this does, because it takes a pilot, it's a sinusoidal, let's see, A or La in other languages, note, and it sort of cuts it so that it gives several harmonics around it and it becomes much richer. The way you can see that is the pilot and the harmonic is by doing a Fourier transform or something similar on that. You see the frequency spectrum, basically, using this kind of stuff. You look at the time-dependent signal, and you turn it into a frequency model, basically. That's one of the things. But another thing is what we started with, because one of the goals here, the one that I led you into, is trying to count the occurrences. Why would you want to count occurrences? Can you think of any other applications? Well, you're probably looking for the distribution function, a probabilistic function, maybe. Okay, what is the probability of this value occurring? Aha, that you can use this for as well. So that brings us to the probably titleless next slide. Yeah, which is stochastic. I remember them, luckily. I don't always remember things, by the way, as you probably understood. We have this statement here that says we never have infinite empirical or exhaustive experimental information for validating our models. We never do. I mean, the nature has started, God knows when, if God knows if there is God, if that is how it all started. We don't know that either. So, but there's no beginning and there's no end that we can see. So we can never really complete something through. So what we need is empirical approaches often. We look at values and we try to guess what it may be, whatever it is. And we try to estimate values, predict values, and that kind of stuff using random or stochastic techniques, basically. And our distribution function from Fourier transforms come in handy in this case of stuff, because the distribution function basically says something about the probability of some value, or a variable being less than a value or more than a value, whatever, ranges. Okay? Statistics. Okay, you know this. This is average. The mean or the average is just you add all of these and you get the total value and divide it into the number of them, and then you get the average, basically. That's very good. But it's not, it models something. It models where your values are around, more or less, most of the time, but it's not enough. So what you would probably do is do things like curve fitting in statistics, basically. And how do you fit a curve? You try to guess the minimize the distance from a postulated curve, it could be linear or nonlinear curve, to the data points, basically, and you minimize that distance, whichever curve it gives you the minimum value, that then you get the curve, and it can be something like this, basically. And for that, you use these values from the mean, basically, which is, well, you probably heard the concept of standard deviation or variance. How much does my point or my estimated point vary from the actual points? Basically, that kind of stuff. And then you have another level of modeling there. But there are many others. There may be values that are obviously too far away, which would be what we would call anomalies today. Anomaly detection is what we talk about. Those are the outliers. Those are the anomalies, basically. And you would have other things, like if you have groups that seem to gather around one average value, that would be, and maybe there are several of these groups around different values, that would be k-means. You probably heard of that kind of stuff. And then you would, using that kind of stuff, you can also do clustering or grouping of values and all that stuff, so that you sort of try to guess what nature is trying to tell you, modeled and the stuff empirically, basically. So that's a little bit about stochastic modeling, but watch out. This is very interesting because you have this curve fitting and all that stuff. And let me see. Well, before we go to the AI part, which is the last part, I'd like to give you another tiny story. It's not going to be very fast, this one, I'm sorry. The other story is when I came to Norway, I came to work with a lady called Ellen Histal. Many people probably have never heard of her. They were working with Kristen Nygaard and all the other people, but she was from statistics, the Department of Statistics of the University of Oslo. And her project was trying to figure out, trying to interpret handwriting, read handwritten text. So it was, in a sense, pattern recognition of some sorts. Okay, and what the technique she used was one of the she was one of the first, because she knew the inventor of the technique. Zadeh is his name. Instead of using probability, she used possibility. The difference between probability and possibility is teeny, but important. Probability is based upon set theory, where an entity, that a thing is either a member of a set or not. So what you're trying to do in probability is figuring out whether it's a member of something or not. But that doesn't help in certain kinds of estimations. Well, how would you call it? I mean, you can't really talk about beauty or good or bad. Those are pretty fuzzy concepts. So they're not really either this or that. There are lots of gray zones in nature, and you can't talk about it. And handwriting is a little bit like that. If you look at an A, so someone has written an A, a capital A, pretty fast. The line will be slightly curved. And then the line across may either cross and be outside the two lines, or maybe not connected to one part. So is that an H? Is that an A? You don't know. So that kind of stuff, what we write, we use our cognitive skills to read, basically. So it's not only mechanical interpretations. So that's one of the things that she tried to implement in a project with some French guys again. I don't know why I know so many French people, but that's my destiny, I suppose. They used something called the fuzzy set theory created by Zadeh. And the difference between set theory and fuzzy set theory is that things in fuzzy set theory have a membership degree. In a sense, probability says if something is 100% in a set or 0% in a set. This says, well, a thing can be 70% in a set and 30% not, basically. So it adds this concept of membership degree in sets and builds upon that the possibility theory. This is another dimension of stochastic modeling. Right. Now, the title is Artificial Intelligence and Machine Learning. We've come to the last part, basically. None of the concepts are new, except that they have been on and off. It's called the winter of AI, usually. It has experienced a winter, artificial intelligence has experienced winter twice, not only once. As you can see, in 70 to 80, it was a long period, and then it came back again with expert systems especially, and Lisp became very in. It's a programming language, it's Lisp-oriented programming language. It started to become very in again, but in both cases, it failed. It failed to meet the expectations. The expectations were very high. That's one of the reasons. And one of the reasons that it failed to meet the expectations is because computing power wasn't enough, basically. And the algorithms were a bit so-so. The algorithms have changed a little bit, not that much. They've always been around, but still there has been improvements. And the biggest improvement is in computing power. One of the reasons is because remember all these data points and Fourier transforms and God. Have you tried to calculate the Fourier transform of something on a computer? Say 1 gigabyte of data? Okay, you can't. It will take you, well, I don't know, you will be pretty old by the time it's finished. And so the world will have moved on. So one of the reasons, that's one of the reasons why we have something called the fast Fourier transform, which is for computing, basically. But the idea behind this is that we need computational power for these kinds of things, basically. So computational power, without proper computational power, AI may flop again. There are many people who are talking about that these days. So, but anyway, AI is very much built upon this idea of modeling something, modeling nature, using some sort of an approach. And there are several approaches that has been used. In many ways, the ones that have stayed around are the ones that we're talking about these days are what we call the biologically inspired techniques. And those are the two major techniques that one of them you hear a lot about, and that's neural networks that mimics the neural network, basically. And the other one is evolutionary algorithms, which mimics the survival of the fittest kind of approach, where you have a data set, basically, that gives you a result, and you check the result. It fails. So that DNA dies out, basically. The ones that succeed to a certain degree, they live on and they gather together and become better and better and better, and the other ones die. So that's the idea behind evolutionary algorithms. So those are the two approaches, and both of them are based on mathematical techniques and statistical techniques, basically. Both. And one of the nice things, I think it should be here, one of the nice things that we should not forget, one of the simplest first slides in statistics, extrapolation. I mean, you fit a curve, and then you go follow the curve to the point where you need to predict something. At this time, you read the value. That is also prediction, guys. We still use it, believe it or not. It's one of the simplest forms of artificial intelligence. I don't know whether you would call it artificial intelligence, but I do, basically. So this last one is going to be called, the title is AI ML, What's Next? One of the things, if you're interested in this kind of area, in modeling in general, and especially in artificial intelligence or these kinds of approaches, stochastic approaches and like that, one thing that's happening slowly, pretty slowly, you don't hear much about it, is using old algorithms, known algorithms, and mixing them. So mainly because a certain approach has advantages and disadvantages. So each one of them has their advantages. So one of the things that we want to try to do is reduce the disadvantages by putting them together, removing some of the disadvantages, and amplifying the advantages of the techniques. So one area that's very interesting, because it relates to the kind of my biological science kind of part. I have many departments in my brain, and one part is biology, molecular biology especially. What if we mix the steps of these two things, this evolutionary algorithm and neural network, and try to use a neural network as a series of data where the in between weights as the sort of DNA, if you want, initial DNA, and then see if it succeeds, then kill it if it doesn't, or improve it. So using these two kinds of approaches together, one of this is just an example, there are many other examples, but this is also happening, and if it doesn't happen, AI will flop again. It needs to improve, basically. But there are other areas as well. One of the problems with these neural networks that we do research on in this little company of ours, as I mentioned, is explainable AI, because the neural network doesn't tell you how it came to that result. Basically, it just gives you a result. You don't know why it has given. So that doesn't give the human user trust enough. Sometimes it's not that important, but in the area, in biotechnology that we work, it is important, because you can't just pop out drugs and say, okay, my AI told me so. Why? So you need to have the control over why this has happened as it has. That area is called explainable AI. Okay, and this is just fun. The 3 laws of robotics, because AI isn't only a technique. AI has a series challenges related to ethics and how safe it is. If we start creating, people have been talking about robots that will kill all of us, take over, that kind of stuff. So I'll just put this up to remind you that AI isn't just a technique, it's not just one kind of science. And surprisingly, this Mr. Science fiction writer, Mr. Asimov, he came up with this kind of worry and wrote a book called I, Robot, a series of books, actually two books, as creating these three laws of robotics. A robot may not injure a human being or through inaction allow a human being to come to harm, but then the robot shall protect itself as well, that kind of stuff. It's sort of a hierarchical set of principles that is supposed to govern the robot. But it leads to something very interesting, another next step. How are you going to make sure that this robot is functioning? And if it's not, why not? This is going to be relatively complex. He came up with robot psychiatrist in his, it's implied. In a sense, the robot psychiatrist was a good code reader who read the code and figured out what was going wrong inside the robot, basically. And that is probably going to be, if we're going to go in that direction, that's probably going to be a necessity to understand. There are areas like code verification and that kind of stuff, but we will need to, again, like the statistical case and all that stuff, we can't do it by hand, so this needs to be automated to some degree. We need some maths, some technique, some algorithms to do this as well. And that is probably going to be an important one in the near future. So thank you. If you like this kind of stuff and just to take a beer and talk about this kind of stuff, my email is available. Just mail me. You're welcome to talk, ask, and all that stuff. Thank you very much.
Do you have time for questions? Time for questions?
I don't know. Not really. I talk too much already.
So, okay. So thank you very much. Unfortunately, we don't have time for questions, but I guess that if you have any, you can just write.
Yes, you can always write to me.
Contact him.
My
Yeah.
mail is available, and I try to answer.
Thank you very much. Thank you very much for being here for your presentation. We will have a few minutes pause now, and then we will go on with the next speaker. Thank you.
Actually, you just need to, sorry. You just need
Again? For just, yeah.
Yes?
Okay. So, oh, sorry.
Close. I got.
We're ready. We're ready to start again. So I want to welcome. Yes, I know who she is, but still.
The rest, barely not.
I want to welcome Ilse. Ilse is also has been one of the volunteers in Pint of Science for 2 years. So, and she did a great job, really great job. So thank you in advance also for this. Ilse tonight will present, of course, about her work, but she will also introduce Simula. As I said before, Simula is our sponsor and co-host for today's sponsor for Pint of Science, the festival this year, and it has also been our sponsor in our previous editions. So Ilse is a biophysicist and a postdoctoral fellow at Similar Research Laboratory. She finished her PhD last year on experimental and computational model on the small conductance calcium-activated potassium channel. She continued her interdisciplinary research in biomedical technology to develop models for drug testing in virtual populations. So, Ilse, please, the stage is yours, and thank you again.
Thanks. Thank you. Magic. All right, so as Lavinia mentioned, I will talk about my work, but first we will get another introduction of Simula, which in this case is not the language, but is the company or research institute, actually. So Simula has been sponsoring Pint of Science since it started in Norway about 3 years ago. I may have something to do with that. But Simula's vision is, and I'm just going to read this because, yeah. Simula's vision is to be an excellent research lab, solving important and fundamental problems of science and engineering and training highly qualified experts and leaders and developing commercial enterprises. So to understand Simula a little bit more, Simula is a research laboratory. It is kind of in between a company and a university, more leaning towards the company side of things. And all of the work done at Simula revolves around the ICT sector, so information and communication technology. Simula has 5 fields, and mainly revolving around scientific computing, software engineering, communication systems, machine learning, and cryptography. With education as the main component, that being at any time, we usually have around 45 master students, around 52 PhD candidates, and usually around 47, I think at the moment, around that number, postdocs, of which I am one of. But in addition to that, Simula is also focusing on innovation. So Simula also has a incubator for tech entrepreneurship and invests in startup companies and is consulting in deep tech and R&D. So some of the research that Simula is working on machine learning is, for example, focusing on deep neural networks and its application in that field in specific. Another one, as an example, is communication, where Simula has many projects in IoT and 5G infrastructure and focuses a lot on research in sensor networks and signal processing. And then the department that I'm part of, scientific computing, which is focusing heavily on biomedical modeling, of which we'll hear 2 talks today by me and Marie. In addition to sponsoring Pint of Science, Simula is also sponsoring other events like the International Mathematics Olympiad in July this year. And in this Olympiad, there's about 600 international students that I think high school students that are coming to Oslo to solve mathematical problems and hopefully get interested for a career in STEM and maybe even at Simula. So why does Simula sponsor all of these events like Pint of Science, like the Olympiad? So Simula really has a mission to encourage people to go into STEM, to get excited for the research, and wants to help with that, with encouraging this field, and of course, hopefully also recruit some of the best of that. That was the end of at least the Simula part. I'm just lazy. It's your job, isn't it?
No.
It's your volunteer job, isn't it?
I started it.
Yeah, exactly. Can't blame you for this.
Yeah.
All right. So now to the more exciting part. I will talk about the heart in my computer because I actually have multiple of them in my computer. Why do I have a heart in my computer? Yeah. Let's see. So I will answer 3 questions during this talk. First of all, why? Second of all, how? Third of all, what can we actually do with this? Let's start with the why. So why do we want to simulate a heart? So cardiovascular disease is actually the #1 cause of death in the world. However, a lot of the testing and understanding of developing drugs or understanding the disease itself relies heavily on animal experiments, on bench tests, laboratory work, and clinical trials. However, if you look at the clinical trials, there's actually, so that's kind of the last step before admitting a new drug to the market, they're very costly and they take very long. They can take up to years just to develop a single drug or treatment. And even then, the drug approval rate is very low. So a lot of companies invest money, time, and research into what kind of drug to develop. However, at the end, it might never make it to the market. So how can we make this any better? Laboratory experiments are very useful, but they are done on cells, on animals, on not humans. In addition to that, a lot of the bench tests, they take time, but they are also based on models of models. So if we have a cardiovascular disease that is represented in a mouse, might not exactly be the same as in a human, even though we can extrapolate from that. So the part of work that I'm working on in SimCardioTest, in my case, which is a project from the EU, is to actually improve these clinical trials. So now that we know a little bit about the why, how, which will be the main part. So most of my day looks like this. There's a lot of modeling and simulation, which sounds very fancy, but there's also a lot of debugging. It's a lot of looking at code, but what does the code actually mean? And to understand that, we need to know a little bit more about the biology behind it. So let's get back there and start with the heart. So the heart has 4 chambers, 2 atria at the top and 2 ventricles underneath that. The entire function of the heart, most known to most people, is that it will pump the blood through the body. But it does this in a very coordinated fashion. So first the atria contract, pushing all the blood to the ventricle, and then the ventricle contract, pushing all the blood out of the heart into the body. But this has to be a very well-timed connection, because if the atria are not empty yet, then you might get less blood, less oxygen in your bloodstream. So to do this, the heart has an electrical conduction system, and that basically starts with an electrical signal at the top of the atria, spreading through both of the atria and then spreading to both of the ventricles, causing the entire contraction. All right, so we have an electrical system, but to understand a little bit more about how we can simulate that, we need to look at the cells in the heart, the cardiomyocytes, or more precisely, the membrane of a cardiomyocyte. Because the membrane of a cardiomyocyte, or the membrane of the cardiomyocyte, is basically what we simulate often in the models. But to understand that, if we have a compartment that is closed off, but sodium, in this case, can travel through it, then nature will try to avoid any chaos and look for the equilibrium and moving the sodium ions into the compartment to kind of equalize the distribution on both sides. However, in a cardiac cell, this doesn't happen. The membrane is not that permeable, and ions cannot just move back and forth as they please. So this doesn't happen. Luckily, we do have a system in place, which are called ion channels. And these ion channels can, in the right circumstances, actually allow some of the sodium channels to pass through. Then they will create somewhat of a balance or equilibrium. However, these ion channels are very specific about when they open, when they don't open. And that basically gives us the timing and the different steps in the action potential or in the shape or the contraction of the heart. So in addition to having only opening at very specific times, there's also a large variety of channels of pumps and other processes. So you can see that even though the ion channel is open, the sodium did not distribute equally, because there's a lot of other processes going on that will maintain or prevent that from happening. But if we then look at the difference, so we do have a difference between how much positive charge we have on the inside of the membrane and outside of the membrane, which continuously changes over time. And with that, we get an action potential, for which in this case, the change of the sodium channels will actually cause the potential to go up, so from a negative voltage to a positive voltage, and then back down with some of the other ion channels. So this has to be timed very well, but luckily we can measure this in experiments. So we can see when an ion channel is open, when it is green, or when it is close, when it is in red. We can then measure exactly how long it stays open or closed, in what conditions it will open, when it will close, maybe if it reaches a different state that we didn't know existed yet. But all of this is experimental work. But if we know when a channel is open or closed, we know what state it is in, we can represent this as a Markov model, where we basically have one state that says the channel is closed and one state says the channel is open. We can connect those with rates because they will switch between the two, as we've just seen. So we can change, or we can get a parameter, alpha and beta, to kind of identify how often will it change or how fast will it change. And then we can list those as differential equations, as we've already seen a couple before. That really helps. But at least we can see the change of how likely it is that a channel will go from closed to open or the other way around, depending on those parameters, alpha and beta. However, alpha and beta are not really something we can calculate. Alpha and beta are letters, not numbers. So how do we find what numbers should go there to actually represent what the ion channel does? So in that case, we use an optimization. We add the shape or the state of the model, how it should look like, which parameters we want to optimize, and some of the experimental data. Then put that into an optimization, in this case for the parameters alpha and beta. Within that optimization, we pick a target, or we pick a set that would kind of work based on what we know from experimental data or any other input. Put that in a computer, simulate it, and then check if the simulation matches the experimental data or not. If it doesn't, we keep going. We check another parameter set, go through the same loop again. The computer does, I don't. Go through the same loop again until we reach a point where it does actually equalize, where we do have a simulation result that matches the experiment, and then we know which parameters we should use. Alpha and beta are not just abstract parameters anymore, they're actually numbers we can calculate. Sometimes, though, we never really reach this state. We never really get to anything that resembles the experimental data. However, that might also just mean that we're misunderstanding something in the biology. So instead of this simple model, we might need a more complex one. Yeah. So now we know how we can simulate an ion channel. Of course, we have a collection of those. I have to apply this for all of them together. We know that this then results at the cell level in an action potential, in a voltage that we can measure. But then we can also connect those cells together and actually create the entire tissue. Videos are always the fun part. So then we can actually simulate how the electrical signal propagates through the entire tissue of the heart. So now we know a little bit about how. And now we get to a problem, because what we can do with that is a lot. And unfortunately, I do not have time to talk here for hours. So I tried to narrow it down to something a little bit fitting 9 boxes. Feel free to ask questions about this later, because this will be very brief. So one of the things that we looked at before, right, is that we can look at this mechanistic understanding. We can look at, does an ion channel behave in a way that we thought it would? Or do we need something more complex to describe it? Is there something we're missing we didn't know yet before from experiments or otherwise? We can look at intracellular messengers. So how do different ion channels talk to each other? How do they depend on each other's effects? Of course, we can simulate drug effects. If we know how a drug works, we can put it in one of the equations, see how it alters the equation, and simulate what it does at the cell level. We can translate what's been measured in animal models or in cell lines to what actually happens in a human model. And of course, a computational model is still a model, but I do beg to argue that a computational model of a human might still be closer than a cardiac model in a mouse. We can look at the multi-scale effect. So if we have an effect that we know affects an ion channel, we can actually propagate that and figure out in a simulation how it works in a cell or even in the entire tissue. We can look at different populations that might not be feasible. If we know that a drug works in a certain way in a healthy population, here in blue, then it might be a completely different effect that we have in a population with heart failure. However, we might not have reason to try it in humans and set up a clinical trial, which is expensive and long. So running it here would be easier. We can look at the mechanics of the heart. So of course, we know that pumping of the heart is the main function. So if we look at how the heart actually uses that electrical signal to contract and actually pump the blood out, and if that is affected in different situations. We can look at underrepresented populations, which is a collaboration with the university in Valencia, working on the differences between men and women. Very often clinical trials look at mainly men. Why do different drug effects affect women differently? How can we see that, maybe we need a lower dose for women or the opposite way? And last but not least, the project I'm currently mainly working on is creating these in silico trials. So instead of doing an entire drug test from the lab through animal models, through humans in clinical trials, can we actually predict what happens in a clinical trial before we even do it? To know which drugs we should focus on or which drugs a company should focus on to save a lot of time and money, but also to actually make the drugs a lot better, to make the drugs a lot more useful for people or more applicable to your specific situation. And I want to leave it at that because I think that was enough on the level of detail. So thank you for your attention, and please let me know if you have any questions.
Thank you. Thank you very much, Ilse. And I think we have time for questions. So if you have questions, this is the right moment. Don't be shy. Yeah.
Yeah, the cable's not. You need both.
Yeah.
Online and in the room.
Oh, perfect. No worries. Just a quick question around how do you decide at what point do you figure out if during an experiment data is being overfit and that you have to discard that particular model or run more iterations against the experimental data and the simulated results?
Yeah. Guess I'm waiting for the microphone. Yeah, thanks. So usually you set a threshold. So you figure if you're closer than 5 or 10% from your experimental data, then you assume that it is a good enough fit. But very often, this does account for some visual inspection. You look at the data, the simulated data, you look at the experimental data and think, this is not really close enough where we thought it would be. So not all of it is an automated process. But also keep in mind that most of the experimental data is not as straightforward. Experimental data has a mean and a standard deviation. And as we've heard before, that can still have quite some variation in there. So that also has to be taken into account how to compensate for that. So usually checking if it's within the range of experimental data should give you a pretty good guess.
Other questions?
I don't think your cable.
Here.
Oh, I have two microphones. Hi. Has your model shown up something really surprising that you we didn't know from human observations or animal experiments, for example? And if so, what was it?
Yeah, so the model that I showed before as the example of where we might need, that was too far, where we might need a more complicated model, right? Get there. I like animations, sorry. So in this case, this is actually from the result of my PhD and the small conductance calcium activated potassium channel that was mentioned before, where we actually looked at a very simple model, something like this, or maybe having 5 states, for example, actually being one of these models, so only the top part in the top box. So this is what the data told us. This is what most of this type of data looked like. And we could describe all of that data with one part of this model. However, running simulations by combining 2 different sources of data, so at the single channel level and at the cell level, actually told us that that's not the whole truth. So even though we thought we could describe it with the one data set, we did figure out that we actually need both of these, where the transitions between here are very important and are actually affected by some of the drugs. So we would not have known that, we would not have known the importance of those if we would not have tried to combine 2 different data sets and realized that we get to this cross where it's not working and try again. So I think that for me was one of the things where it's very interesting to see how you combine different things and what you can actually learn from that and what an impact that it has later on.
The next question was, yeah, I'm not sure I can.
I don't think, or just come here.
Just to have a sense of the scale, how many channels are more or less on one cell? Because for the whole Earth, it looks like a lot of channels.
Yeah, so how many channels or how many different types of channels? Exactly.
How many channels?
How many channels? I do not know, millions. How many channels? I really don't know. How many types of channels? There are, it depends on the model. It depends on if you look at a ventricle or an atria. I think there's usually around 10 to 15 different types of ion channels, but all of those are very changeable in how much they are, how much they're present in the membrane, because even having like a repeated activity can change the fact if a channel is in the membrane and therefore functioning, or if the channel is somewhere in the cell floating around waiting to be activated. So the amount of channels in an actual cell is changing rapidly, but it's very high.
Other questions? Yes. Of course.
So just for the public, I'm modeling on the lower scale than the molecular level. So my question is, what we have is, can you generate the model on what about personalized medicine? My ethical question is like, when we have like only human simulation, on when do you think then the population will be ready for that?
Sorry, when the?
When do you think the population will agree to just test drugs that has just been tested only for new in silico trials?
So 2 answers, because, oh. Sorry. When the population is ready and when the authorities are ready are 2 different things. When the authorities are ready is a big part of my project right now in trying to figure out how to get approval from the authorities to actually use a computer, which is rather unfamiliar to most people that are relatively old that work there. Generally, at least the ones that I met. So they're not against simulations. They see the power of this, but they do not have any way of assessing how good it is. Where for experiments, they've been working with it for years, so they know. The clinical trial has an outcome, and they know what to do with that. I think for the authorities, it's a going process, and I think in a couple of years there will definitely be some parts of clinical trials that can be used in silico. And I think as soon as the authorities approve it and it has been tested thoroughly, which is necessary for the authorities anyway, I do think that adds to the convincing power to the population, because if it has been approved, there's a lot of people that would accept it as being useful. And at least for us, the goal is not to replace clinical trials. The goal is to make them more efficient and make them more applicable. So I don't think in any time soon we're going to have to replace clinical trials or animal testing, but we can reduce them and we can refine it by trying to simulate beforehand what we can expect.
Anyone else? Okay. So thank you, Ilse. Thank you very much. And we can have a very small part now, and then the last speaker. Thank you. Okay. Okay. So we are ready to. Thank you. We are ready for our third and last speaker for tonight, Maria. Maria is a student at SimulaMet working on extracting information from a complex data sources. She's also known as this is also known as data mining, and it means that she enjoys looking for underlying patterns behind collection of numbers. Maria has a master's degree in applied mathematics from the University of Oslo and is currently in the final year of her PhD at Simula Metropolitan Center for Digital Engineering. So Maria, please, the stage is yours, and thank you very much for being here tonight.
Thank you. This neuroscientist has collected images of brain signals, and her mission is incredibly important. To understand what happens inside the brain of people with severe mental disorders. And to accomplish this, she has to uncover what lies behind these signals. What kind of activity in the brain produces precisely these signals? And can we extract any differences between healthy people and people with severe mental disorders? I wish that I could stand here in front of you now and say that this is me and talk a lot about the brain. But I'm not a neuroscientist. And my research is not about brains, at least not directly. My research is about the tools in her tool belt. Excuse me. Is this not responding?
The pointer.
No, the mouse, the screen. Is it because of the stream? Oh yeah. Thank you. My research is about the tools in her tool belt. And what do I mean by tools? What kind of tools does a neuroscientist need? Well, let's look a little closer at these brain signals. If we zoom in, we see that they are really just numbers. All data is actually just numbers. And how do we go from numbers to knowledge? Through mathematics. The tools are mathematics. And such mathematical tools or models are essential for many areas of science. Just like a woodworker needs the right tools to build new furniture, a scientist needs the right tool to build new knowledge. And my research, the goal of my research is to make sure that the neuroscientists and other scientists have the right tool in their tool belt. I'm a mathematician or a data scientist. And more specifically, I work with what I consider to be the most exciting and most practical form of mathematics. That's right, factorization. With factorization, I can uncover the hidden truth behind numbers. For example, with the help of factorization, I can reveal to everyone sitting in the room tonight that the number 7 is actually 2 times 3 plus 1 times 1. Okay, I sense that not absolutely everyone in the room are fully convinced of how exciting it is with numbers that are multiplied together. But what if the number is a measurement? A piece of data that says something about how active this region of my brain is right this moment. Then the factorization tells us something about what lies behind this activation. And what if I measure this for all the brains sitting in this room? Then I will get a row of numbers, a vector. And factorization on this vector reveals something about how we are all different right here in this region of the brain. And what if I don't stop with just one measurement? What if I measure several regions all over the brain? That will give me one row of numbers for each brain region, so a whole table or a matrix. And factorization of this matrix, that gives us the patterns behind the measurements. And this is a little easier to see if we arrange the numbers after their position in the brain. Then we see that these numbers, they form low-resolution brains. And all this data can be factorized into two sources. The first source, the one to the left, reveals a network of activation in regions of the brain that's related to motion and to vision. And the second source, the one to the right, reveals activation related to the senses. And the numbers that you see on top, they tell us how much of each of these sources are mixed into each person's brain signals. This is why neuroscientists use factorization, to separate out these networks of brain activity and to extract some insight into how these activity patterns change from person to person. So this collection of signals is like a recording of classical music, which is made up of many different bands, many different instruments, a whole orchestra of instruments that create many different sounds that are mixed together into the complex and beautiful music that we can experience. A factorization is about separating out these sources, which instruments are playing, and which notes they are playing, and how much we can hear from each instrument. These kinds of tools are often called matrix factorization, and they're useful for more than just neuroscience. Netflix can, for example, use factorization on a table of likes and dislikes given by a group of people, such as yourself, to a list of movies and television shows, and extract movie genres and how much each person enjoys each genre. So matrix factorization tools are incredibly useful to extract patterns from data tables with two dimensions, such as people and movie, or people and brain region, like we saw earlier. But what if the data you gathered has more than two dimensions? And what if the third dimension is time? So the neuroscientists that I collaborate with, they have recently gotten interested in so-called dynamic connectivity analysis. And this means that instead of just looking at the one image, we look at several images, like a movie. Another question is, what happens with these underlying networks of activity in the brain over time? And how can we account for this change when we don't even know what type of change it is? Well, what we do classically in mathematics is that we just assume there is no change at all. Or we are a little more generous and assume only linear change, which means that things can change in a straight line up or a straight line down. And in practice, for the neuroscientists who have to actually use the maths, this means that these underlying networks of activity, they can only change in signal strength. And what if this is not a case? What if the second violin in the orchestra not only changes the volume they're playing at, but also which notes they're playing? What if the underlying networks of brain activity not only changes signal strength, but network structure? Analysis of such changes can give new, valuable insight into brain activity. But we'll never know if we don't have the tools to capture these changes. If all you have is a hammer, then every problem looks like a nail, and you'll never screw in a single screw. If you force your data into a linear model, you will get linear patterns out. Nothing more, nothing less. And that's where I come into the picture. I work with factorization tools for time data. Tools that can capture more than just straight lines. And to do this, I need a combination of mathematics and programming. To create a new tool, I start with something that already exists, preferably something that is already done well on a slightly different problem. And then I analyze this tool really closely, see what kind of strengths and weaknesses I can find, and what kind of improvements I can make to create a tool that's even better. And then I pick up the computer and I use programming to build prototypes of the tools and stress test them. Really push them to their limits. With programming, I can simulate my own networks of brain activation that I mix together to create fake brain signals. And then I can see if the factorization tools manages to separate them again correctly. Here are two of my simulated networks. And as you can see, I've made them dynamic. They change over time. And then I use these as the truth, mix them together. And with the classical linear tool, I can extract these patterns. And here we haven't really captured the dynamics at all. And the activation patterns, they don't even show activation at the right places. So this is a failure. And with my new modified tool, we can extract these patterns. So here we have both the correct activation patterns and how they change over time. I love numbers. And I love making things that are useful for other people. So sitting in front of the computer and creating factorization tools that can help neuroscientists improve their research, that's just the perfect job for me. And when I'm finished with the prototyping and the testing, well, it's time to look at the real thing. So these are images from an experiment where the participants, which included healthy participants as well as patients with schizophrenia, they were equipped with a headset and then instructed to listen and push a button when they heard a sound. And from this dataset, with my new modified tool, we can extract these patterns. Again, we see two sources. And the first source reveals activation in an area known as the sensory motor cortex, which is related to the senses and to motion. And the second source reveals activation in, among others, the auditory cortex, which is related to sound. So basically, these patterns are very relevant to the task of listening and pushing. And one observation that the neuroscientists were interested in is these patterns, while they are present in both groups, they are a little stronger for the healthy patients, which could, for example, indicate that they might have had a little bit of an easier time concentrating fully on the task. And the other exciting thing is that these patterns are dynamic. They change. And they change more than just signal strength. So with a classical linear tool, we could never even look at this change. And by creating a new tool for factorization, we have opened the door for the dynamic analysis of brain signals. Thank you. Any questions?
Okay. Thank you very much, Maria. Do we have any questions? Yes.
Very stupid question.
We don't have stupid questions, please. If you can come closer a little bit. Thank you.
It's a very dumb question. You said you love numbers. Which is your favorite?
Should I? I think it's between 0 and 1. Excellent question.
Other questions?
So is this very specific to the functional MRI data, or can it also be applied to things like the EEG, for example, or even cardiac? Because there's also a lot of the echo measurements that are done in cardiac that I think are based on somewhat the same principles.
Yeah, this is a very good question. No, this is not specific to fMRI data at all, really. Like I said, I'm not a neuroscientist. I'm a mathematician. But I collaborate with neuroscientists, which is why I've looked at this data. But it can be used for other data as well. I look specifically at data that has, it's sort of, if it's a cube, and one of the dimensions are time, then these methods fit very neatly. So right now, for example, I'm looking at planks, just wooden planks, a movie of planks, and that's also interesting to me.
Look at the cardiac data.
Yeah, I know. Some people have looked into that. So.
Anyone else? Yes. Why I ask?
No, but nice presentation. I just have a question regarding. Ask 3 minutes on at the same time. At the 3 minutes. No, it's like, how can you use this model? I think it's like a big with Ilse's project or what she presented. You are fitting a model to expand data. What do you need to get it as a predictive tool in the actual like medical environment? What do you need to get that? Is it a better model, more data, or like a more general model? What do you need? Or when we can see that to have such tool as a like medical diagnostic?
So these types of methods are usually called unsupervised machine learning. So I'm not that interested in prediction. I mean, you can use these for prediction sort of later, but I'm more interested in exploring what's inside data. Like you have a data set, and you know there's some patterns in there, and you want to look at them. It's more about decomposing than prediction. But of course, more data helps. But the nice thing about these methods that they work if you have smaller data sets as well. You don't need as big of a data set as you need for a neural network, for example. But more data is always good. And other things that you could do is apply domain knowledge as regularization to your optimization. That's usually how, that's what I've looked a lot into for improving and making the component more interpretable, because that's the goal. Like, I want what I get out to be useful for a neuroscientist, to be easy for them to interpret and use further in their analysis. So if I can use something that we know about how the components should look to make them look more interpretable, for example, that's very valuable. That will make the model better.
Other questions? Yes.
Just a follow-up.
Yeah. So when you search that you simulate at the cell level, you analyze at the organ level. If there's any project to actually incorporate both scale.
Like the whole body, the connections
Oh,
between the
yeah,
organs.
Yeah, I mean, my simulations are different because I don't really simulate using physics, because I just look at data. So I simulate more using statistical properties of the data, like type of randomness. But of course, there are definitely possibilities of using simulations to generate data that I then can do analysis on. So
I think also the opposite way, there's a lot of data that we have in, for example, 3D echo of a heart, but we cannot make mathematical sense of it to use it as input for a simulation, or to use it as input to parameterize a simulation. So I think in both ways, you can definitely use it.
yeah. Very good question.
Yes.
Hi, really nice talk. I was just wondering if your method is very sensitive to noisy data, does it then struggle to pick out like the individual signals if the data set is too noisy? Does it have to be like very clean signals of the different factors?
Oh yeah, that's a very good question. Yeah, for my data, it's actually very noisy. It's super noisy because the way it's measured is it looks at how much oxygen is in the blood and uses that sort of to tell how active that area of the brain is. But that is, of course, not a super clear image. So there is a lot of noise, but it still works. But of course, this will depend on the domain and also the noise. If the noise has a weird structure, it will be more difficult. But that's where what I mentioned earlier also comes in. If I have some knowledge about how things should be, for example, one thing that I've looked at a lot with the fMRI data that I didn't really include here is we know that this part of the brain, if this is active, then the one next to it is probably also active. And I can use that to counteract some of the noise and make the result less noisy. Thank you for your question. I've thought a lot about noise, so I'm glad you brought it up.
Yes. Yeah, I need. Yes.
We need the wireless network.
Yes.
Yeah.
Okay, well, that's a standard mathematical question. How much of this is generalizable to higher dimensions? So more than, let's say, 3 dimensions n time, or more than that n time.
Oh, everything. You could use tensor methods on, fourth order, fifth order, whatever, really. And the model that I've been looking at, if I had more dimensions, that would actually make life easier for me. It would be easier to get in something we call uniqueness, which means that the model is stable. It's actually the fact that they only have 3 dimensions that's the main challenge. That makes it a little trickier. But that's a very good question, yeah.
So for a question, so why the population cannot be the 4th dimension? Is it for to generalize it?
So population, you mean people?
Individuals, yeah.
Yeah, one of the dimensions is actually people. So I have people, and then brain region, and then time. Yeah.
Anyone else? Yes.
Oh, you're good. Thanks for the nice and fun presentation. Just a question regarding generalization again. How do you, and if you do account for the physiological differences, like you mentioned earlier, that there's different activations or different thresholds for activations based on some people who have early onset dementia, for instance. So how, when designing these tools, how do you accommodate for those kinds of differences? Because, well, that's question one. And then also, how do you or neuroscientists that you work with decide on what would be a good representation to actually portray those differences well, not accurately, but reasonably?
Oh, that's a very good question. And we don't really account for that because we don't really want to. We want to see what patterns we find just from the data itself. Like, what can the data tell us without us putting in anything about what we think about those differences? Because then we might find differences that are new or something that the neuroscientists have not looked into before. So we don't really have anything like that in the model. We just look at the data, and then we see afterwards if the patterns, if there are any differences. But of course, this is also something like we could encode some of that knowledge in, similar to, as I said, with you could use the smoothness of the brain. You could really use any kind of knowledge if you can describe it as some kind of regularization or something. And that could also be interesting. It's just for this project, we didn't even want to say how many groups, because what if there are more diagnoses under the diagnosis? Like, we don't want to put that in there. We want to see what we get out. We want to let the data speak to us.
Thank you. Yes. Sorry. Sorry.
So since I'm coming from computer science, I'm going to go with a very theoretical question about algorithms. So matrix factorization, usually that depends on which kind of numbers you are putting in. But if you're putting a lot of numbers, especially negative numbers, it can be an NP-hard problem. So when it comes to a brain, you are using probably a lot of numbers, and the matrices are huge.
Mm-hmm.
So given the fact that the algorithms for matrix factorizations are very challenging, how do you cope with time and space? And do you ever hit the computational wall? And of course, how do you deal with that?
Yeah, I hit that wall all the time. As you're right, they are very big, at least in one dimension. They are about like 250 on one, and then 14 on one dimension, and then 67,000 on another. And yeah, it takes a while As you say, it's a difficult problem. We try to use as smart optimization as we can. And I haven't looked that much into speeding it up. That's something I'm looking a little bit into now, actually, but it hasn't been a focus yet. I've been focusing on flexibility of the algorithm, being able to put in, like I mentioned, information about smoothness or something like that. So that's the part of the algorithm that I've been tweaking and improving. But there's also another part to look into, and that's speeding things up. Because as you say, this is definitely a big challenge, and I have to run on a cluster and be patient.
So one comment on that, Simula has a department of high performance computing, which I think we both rely on a little bit to try to speed things up and make it more computationally efficient. So that's kind of a lot of research going on by itself in that development as well.
Yeah, absolutely.
Again, other questions? we can do that. So, okay, we're fine. So thank you very much for your contribution. Thank you very much, Maria. And so I think we can go on with the quiz, right? Yes. So we are going to have a quick, not very quick, actually, no, quiz. And Sean is chairing for this. Our new volunteer, actually, in the Pint of Science Oslo. So it's also her welcome in the team.
Oh my God, this is too much.
What? Wait, too many microphones?
Yeah, too many wires.
Can you? Yeah.
Oh, should I attach that to me?
Yeah. No, other side, maybe.
Hello. I can hear my own voice now. No, it's okay. I just hear it from somewhere there.
Yeah.
Okay. What's this for?
That is no.
This is for the streaming.
Okay, but what do I press? Nothing. Okay, nothing. Okay. And then how do I control the screen? This. Okay, so you can, I think most of you know what you're doing, but you just go to menti.com and enter this code at the top of the screen. That will take you to the quiz. Yeah, it looks like most of you have got there already. How am I moving?
Wait.
Okay. But how? On this? No.
Although I just
No, I don't think so. I don't think this one.
It's not on this screen.
It's still connected.
Oh. So if I press enter on this, it will change. Okay. Okay, then we're good. Are people still entering? Okay. Okay. Oh, there's a volcano. I like that. I'm a geologist. So. I don't think you get to choose. No, you just assign. It just knows whatever best matches your personality, I think. Yeah. That might have been offensive to someone over there. Sorry. Okay, yeah, so So the questions are all themed on today's talks. And since today we're doing the quiz at the end of the talks, you should basically know the answers already. Usually we do the quiz at the start before people know what they're doing. But today I'm expecting full marks for everyone. So you are timed as well. So it's not just about getting the right answer. You've also got to be the fastest as well. The faster you are, the more points you get. Okay, so let's begin. Scream now if you've not entered yet. Okay, I think that is a good sign. No, there we go. I suspected so. Yay! Oh, well, it works for you. Yeah, okay.
It's my computer.
Okay, so what is data mining? You have 15 seconds to answer. No, 15 seconds to read the question.
In fact, you're in front of the screen.
Sorry.
Read it on your phone, dude.
Yeah. Yeah, you got it right on your screen in front of you. Okay, there we go. Most people got that one right. That's a good sign. People have been paying attention. So, the arrow. Ah, yay, okay. Right, let's see, leaderboard 1. Who's raced ahead at the start? Oh, it's tight. Oh, nice, okay, F. Very mysterious name, F. Okay, question 2. Answer fast to get more points. What is neuroimaging? Should definitely know this. Ten seconds. Easy, right? We're starting slowly. They'll get more difficult. Yay, there we go. Most people got that. It's not producing a succession of images with your mind. No, sorry. That would be cool, though.
So are these questions already answers about what it actually is, or you want it to be? Because that might.
Yeah, I think we want it to be that, but unfortunately, no. Oh, and now we've got K. The mysterious K is now the fastest. Burger emoji. Great. Oh. Uh-huh. Oh, yes. Okay. How many times does your heart beat in one day? This is a really tough one, I think. People a bit more unsure about this now.
It was not in the presentation.
No. Oh, okay. Finally, we're going to diversify a little bit. Only 9 people got that one. 100,000 times. Your little heart is working hard. Oh no, KF and leans. What were you thinking? Okay, there we go. Aga's taken the top spot. Next question. 412. Stand the other side of this, I think. Which of these are used for drug development? This was in the presentation. I hope no one got too trigger happy there. Yeah, it was all of the above. A couple of people a bit too trigger happy. All of these things are used in drug development. Let's see how that affected the scores. Not too bad. There you go. Everyone's on board with that one. Arga still keeping the top spot. Question 5. Which 2 computer scientists won both the von Neumann Medal and the Turing Award? This was IEEE, there we go. This was very, very briefly skipped over in the first presentation, so you'd be forgiven for missing that one, I think. There we go. Christian Nygaard and Ole Johan Dahl. A bit of trivia for your next pub quiz. Oh, most of these people got that one. Impressive, actually. Maybe it's just me that doesn't know that. Oh, Arga, you're struggling. Oh, no. Okay, question 6. We keep going. Your heart is regulated by what? Five seconds. There we go. Electrical signal. Well, nearly everyone got that one. Sorry, the one person who got contraction there. Sorry.
It wasn't me.
Nice, pretty even. Still all for the taking. Question 7. What is a matrix?
A movie.
Well, it's funny you should say that. I think we're good with this one, right? Yes. Oh no! 2 people got confused between matrix and mattress. Wow. That is unfortunate for you. Oh, we're not changing much anymore, but it's still pretty tight. Question 8. See if this will trip some people up. Symptoms and treatment of heart disease differ between men and women. True or false? Nearly everyone's voted. Oh, I think one person's deliberately getting the wrong answers now. 26 people said true, absolutely. Just a matter of who was fastest then. Whoa, super tight. Kulhuzgugu is doing the best. Don't know if that's an acronym for something. Question 9. We've got to differentiate those top people. Oh, here you go. This is a bit more difficult. What is Goldbach's conjecture? Some people are going to get this one, surely. I think that might have been a bit of a random choice for many people. Wow, okay, there we go. Now we're going to start making some differences. Yeah, every even number greater than 2 is the sum of 2 prime numbers. I don't know. Yeah, apparently that's true. I didn't prove it, but... Oh no, Aga! Goodbye. Kulhugzugugug still clinging on to the top spot. Right, question 10. 3 more questions to go. What does fMRI stand for? Maybe you remember this. Oh, a full house. Yes. Yeah, no one tripping up on that one. Functional magnetic resonance imaging. There we go. Okay, don't expect much to change here then. No. Cool. 2 questions to go. Question 11. See if we can make some in the last 2 questions. When was the term artificial intelligence first coined? Some people thinking hard about this one. Oh, there we go. Yeah, earlier than you think, actually, 1956. I think that might have eliminated some people. Oh, there we go. A and S. I'm sorry. Goodbye. Okay, cool. That's good. See if someone can overtake in the final question. How many times did artificial intelligence have its AI winter? See if you remember this from the first presentation. I'd love it if there's a last-minute overtake. Oh, okay. Most people remembered that, yes, twice. 2 times it was abandoned before becoming the new trendy thing again.
Some were already predicting
Oh no! Course, good. What did you do? Oh my God! Okay. There we go. Oh my gosh, that was a very dramatic finale there. Okay, so in the end, sorry, Kuleskuga, you clung on for so long, but Zimon somewhere on that middle table? Yes, okay. And Zimon gets a free drink. There you go. Come and see us. There you go. Congratulations. And you also get the kudos and respect from everyone in the room as well. So congratulations for that. Yeah, okay, so come see us, Simon, and everyone else. Yeah, thank you for attending. I'll hand back to Lavinia.
Thank you very much. We are at the end. I guess it's pretty late, so I would just remember that our next event is the Pint of Science Festival, our main event. Yeah, almost ready to show it. We will be here on the first day on May the 9th, and we will have another location for the next 2 days. Just check our website and social media and be updated. It will be amazing. It will be great. It will be wonderful. Yeah, it's fine. From May the 9th to the 11th. So please follow us and see you the next time. See you in May. Thank you very much for everyone.